This last section on Chapter 8 is on mutations. And mutations are going to be changes in the DNA. We usually think about mutations as being mistakes. I often call mutations the good, the bad, and the indifferent. Sometimes they're good so that new alleles arise. So if, let's say, there was a mutation and a bacteria now has the ability to be resistant to an antibiotic, it can live in the presence. That's good for the bacteria. Bad news for us, but that's a good mutation for the bacteria. Sometimes be bad, so they can cause the proteins not to function. And sometimes they can be indifferent, where there is a change in genotype, and the genotype is the DNA, but it doesn't change the phenotype or the outcome. And in that case, even though the DNA was changed, it doesn't really make a difference. So oftentimes with mutations, we talk about genotypes and phenotypes. Genotypes are what are determined by the DNA. So if there is a mutation, we can change the genotype. Mutations always change the genotype, but they may or may not change the phenotype. The phenotype is going to be the physical characteristic of an organism. As we saw with proteins, our proteins are made up of amino acids, and the sequence of amino acid is going to be very important for that protein to fold and for it to function. If there was a change in genotype and we ended up with a different codon, if you remember that codon table, we could have 4 codons code for an amino acid. So if we ended up with a mutation that changed the codon, but we still ended up with the same amino acid, we didn't actually change the phenotype. And in that case, the mutation didn't really make a difference. So in this section, we'll talk about the different types of mutations, as well as how we can also study them in bacteria. So we will look at different types of mutations, like a duplication. This is when one nucleotide is repeated. A deletion is when a nucleotide is removed. An inversion is when the segment of the sequence is kind of flipped around. An insertion or translocation, this is when we have a segment of DNA maybe further down the line, and it's just kind of plunked right in the middle of DNA. Breakage is easy to think about because part of it is just lost. And we're going to talk a little bit more in depth about a frame shift mutation. This is when we could have an insertion or a deletion of a single nucleotide, but because of how the codons are read, it actually shifts the frame that the codons are read, and so we could end up with completely different amino acids. So this picture right here is showing the DNA and a point mutation. A point mutation would be a change in the nucleotide. So if you took a look at picture A, so we've got the original DNA, and the code is AAA. And so on our messenger RNA transcript, our codon is UUU. That codon will code for the amino acid phenylalanine. So that is what the amino acid we need in that particular order. If you take a look at picture B, that third A is changed to a T. So we have a base substitution. So now our codon becomes UUA. This codes for leucine. So we're not getting phenylalanine. Is that a big deal? It could be. It could affect how that polypeptide folds. If you see in the third case, in C, that third A is changed to a G. So now our codon is UUC, and we're getting phenylalanine. So even though we changed the genotype, we did not change the phenotype. So that's what those extra codons kind of give us as that little insurance policy. So we can change the genotype. Sometimes it can actually change the phenotype. Sometimes it doesn't, in this case of that third base substitution. So I think it's a little easier to actually think about letters and words. rather than a code. So I've got the original DNA code, but I'm using letters. So up at the very top, A, B, C, D, E, that is our original DNA code. So if we had a duplication and we duplicated that B, now our code is A, B, B, C, D, E. If we have a deletion where we remove the B, now our code is A, C, D, E. And if we have sort of an inversion where we flip around B, C, D, now our code in green, as you see, becomes a D, C, B, E. A translocation or insertion, this is when further down the line, we just kind of take that code of DNA and kind of plunk it right in the middle. So further down in the alphabet, we've got R and S. So as you see with this translocation or this insertion, our code now becomes A, B, R, S, C, D, E. The last one is a little easier to kind of wrap our head around. This is a deletion. So we just kind of take off D and E in that breakage, and now we have that A, B, C as our code. I told you we were going to talk a little bit more about frame shift mutations, and so I think it's a little easier to see with words and a sentence. So this is a silly sentence. The dog ate the cat, but we've got 3-letter words, and those 3 letters represent our codons. Now, this is a silly sentence, but we can at least recognize those words. So we know the dog ate the cat. We can recognize that. If we have a simple deletion in the case of a frame shift mutation, this is just taking one little change. In this case, we're going to delete the E. We could have easily inserted another nucleotide, another letter. But in this case, we're going to delete that E. So with that frame shift mutation, we are shifting how we're reading the frame of those letters. In the case of the protein, we would be shifting how we read those codons. So when we squish these letters together, our sentence, the dog ate the cat, now becomes theta, aga, tet, hack, at. These are nonsense words. They make no sense. In terms of the protein and getting different codons, we can actually get different amino acids. This can have a big consequence on our proteins. So if we took a look at an actual example, if you take a look at the normal DNA code and codons, so up top we've got our DNA code and we're getting codons. These codons are going to get us the sequence of amino acids, phenylalanine, serine, histidine, leucine, and lysine. So in that second block, now what we're going to do is we're going to remove that guanine. So we're going to take out that nucleotide. This is a simple deletion. So now we're going to squish all of those letters together and we're going to get different codons. So we're on track with the first one with phenylalanine, but instead of serine, we're getting arginine. Instead of histidine, we're getting threonine. Instead of leucine, we're getting a stop codon. This is a big problem. So if this polypeptide was supposed to be thousands of amino acids long, we only have 3 amino acids and we end up with a stop codon. So that is what we would call a loss of function mutation. That's bad. No way can that polypeptide fold. No way can that protein function. If we take a look at the third example, now we've got a simple insertion. Now we're inserting a cytosine after that guanine. Again, we're going to push them together. So now we're going to get different codons. We're still on track with phenylalanine. Instead of serine, we're getting valine. Instead of histidine, we're getting alanine. We're back on track with leucine. Instead of lysine, we're getting glutamic acid and so forth. Is that a big deal? It could be, because remember, those amino acids differ a little bit chemically. So as they're sitting in line together, that polypeptide may not fold as it's supposed to with that protein, and that protein may not function. So even though we have a very small change, the way that we shift that frame can actually have big consequences. We can actually study mutations in bacteria. So auxotrophs are bacteria that have acquired mutations that affect their ability to make and use certain nutrients. So we can actually use this to our advantage when we're studying mutations. A normal unmutated bacteria would be what we would call a prototroph. I'm going to talk lastly about a little experiment where we can use an auxotroph to determine whether or not a chemical or drug that we're testing is a mutagen. We know that mutations can spontaneously occur, so there can just be mistakes that happen in the DNA. In DNA replication, we talked about DNA polymerase, and that builds DNA. DNA polymerase has a little proofreading capability where it can kind of change out a nucleotide if it isn't matching, but only to a certain extent. So mutations can spontaneously happen. We also know that mutations can be induced by mutagens. So maybe certain chemicals or radiation, they can cause mutations. So we know that different chemicals and things can also cause mutations, not only in bacteria, but in humans as well. One of the things that we'll talk about when we get into viruses is that sometimes viruses can cause mutations. And we know that there are some viruses that can cause cancer. So here's the thing about bacteria is that if they have DNA damage, if they have mutations, and oftentimes if they're exposed to radiation, these thymine dimers can form where we have these two thymines that are kind of stuck together. Bacteria can be very smart and sneaky and that they can actually repair this damage. And so they can actually take these enzymes, kind of like these molecular scissors, that can actually cut that dimer and then remove them. So they can actually repair some of their damage to a certain extent. That's good news for the bacteria if they have mutations. There were a couple of scientists, Luria Delbruck, that wanted to determine how mutations that allowed resistance to antibiotics arose. And so what they did was they designed a fluctuation test to actually test these two hypotheses. So hypothesis one was that if mutations that cause resistance to antibiotics happen spontaneously, then when they are growing in the absence of the antibiotic, there's going to be a great fluctuation in the number of bacteria that have that resistance mutation. So this is just to say mutations happen spontaneously. Doesn't matter if they're exposed to that antibiotic or not, they're just going to happen. The second hypothesis they had said these mutations for resistance can actually arise only in the presence of the antibiotic. So that bacteria had to be exposed to that antibiotic in order to have that mutation arise. So they did a test. This is what is called the fluctuation test. And we're not going to get too bogged down in the weeds and the detail with this. But what they did was they grew bacteria in individual containers and then in a big flask. The take-home message from this fluctuation test and the results they found proved hypothesis one, that mutations just happen spontaneously, that bacteria don't need to be in the presence of that antibiotic to get those resistance genes from mutations, that they just happen spontaneously. So this is what Luria Delbruck found in their fluctuation test. The last test that I'm going to talk about is the Ames test. And the Ames test, this is actually a toxicology test that determines whether or not chemicals, drugs, whatever it is that a company is trying to put out as a product is a mutagen and causes mutations or can cause cancer. This is a very common toxicological test. And if you were to actually buy a chemical from a supply house, it would actually come with a material data sheet. On that material data sheet, it would talk about the chemical, if it's flammable. And usually the last thing that they talk about is this Ames test, whether or not this chemical causes mutations. This is going to be important if we're working with chemicals in the lab because we want to take some safety precautions. If a pharmaceutical company is putting out a drug, they want to prove that this drug does not cause mutations, does not cause cancer. So with this Ames test, it actually uses an auxotrophic form of Salmonella. Remember, this auxotrophic form has special nutrient requirements. So with this particular auxotrophic Salmonella, it cannot live without the supplement of histidine. It actually has to have histidine, this amino acid, in its media because it cannot make it itself. So in order to grow it, you would need a supplemental media with histidine in order for it to grow. So what we would do in this Ames test is that we would actually take this auxotrophic Salmonella, we would plate it on an agar plate that is lacking that histidine. Now, because this Salmonella would require that histidine in the media, it would actually die on that plate. And so this is how the scientists would test whether or not that chemical or drug they were testing formed mutations. So they would take a filter paper disc, soak it in the chemical that they wanted to test, and then put that on the lawn of growth. Then they would incubate it. If that chemical did not cause mutations, then that bacteria would not be growing, so we would not see growth. If it did cause mutations, then we would see growth. And that means that that particular auxotrophic Salmonella developed mutations to allow it to now make that histidine so that it could live. So if we saw growth, that's a good indication that that chemical caused mutations. So these results of an AIM test would be on that material data sheet so that we would know that we needed to take special precautions because that particular substance was known to cause mutations.